Thank you, Wayne. Our final presenting company this morning is DepYMed, which is a joint venture between Coastman Harbor Labs and NASDAQ-listed OR Pharmaceuticals. DepYMed is developing what I like to call the magic pill that will be an orally taken medication for obesity, diabetes, and HER2 cancer based on trodisquamine and related analogs. Presenting this morning for DepYMed is Dr. Irach Taraporewala.
Thank you very much. Good morning all. Thank you for your attendance and your interest in hearing the DepYMed story. We certainly hope this joint venture with Cold Spring Harbor can burgeon into something successful and serve as a valuable model for what our industry and an academic center like Cold Spring Harbor can do working together. As indicated, we are a joint venture between Cold Spring Harbor Lab and OR Pharmaceutical. OR Pharmaceutical is a New York City-based NASDAQ-listed company. We listed on NASDAQ just 11 months ago. Our main programs in the parent company OR are focused in ophthalmology and oncology, and we have phase 2 clinical trials in cancer cachexia, in wet macular degeneration, diabetic retinopathy, and in ovarian cancer. We did have pipeline compounds which were PTP1B inhibitors, which are protein tyrosine phosphate 1B inhibitors, and the leading research institution for research in that area happens to be Cold Spring Harbor Lab at the lab of Nick Tonks, who is the world-recognized authority and, in fact, the discoverer of the enzyme and its therapeutic potential. OR has been providing compounds to Dr. Tonks and his lab to test over the last 3 years, and this joint venture stems from the discoveries coming out of CSHL's labs. The lead compound is trodisquamine, which is a small molecule allosteric inhibitor of PTP1B enzyme, which is the therapeutic target. We are hoping to enter a phase 1 single agent trial in an oncology application by this year's end. The safety data for this compound had already been demonstrated in another therapeutic indication for up to 65 patients to date. So the lead program, trodisquamine, it is a small molecule selective inhibitor of PTP1B. Unlike other companies' approaches, which is to find active site interacting inhibitors of this enzyme, PTP1B actually does not act at the active site of the enzyme. It binds to a distant allosteric site, modifying the enzyme and rendering it non-functional, and therefore we can get very high selectivity with this type of approach, whereas other PTP1B inhibitors that have been directed at active site structures have the problem of cross-reactivity with other related phosphatases producing side effects. So PTP1B inhibition by trodisquamine is very selective. PTP1B has, over the last few years, become a well-recognized therapeutic target for oncology, obesity, and in diabetes. And the activity has been demonstrated for this compound in various in vivo models. Phase 1 clinical studies have been conducted for obesity and diabetes applications in single and multi-dose escalation studies, and the compound had an acceptable safety profile in those trials. Trodisquamine in oncology is very interesting because PTP1B activates the HER2 pathway. HER2 is the human epidermal growth factor protein that is a known accelerator of tumor growth that induces metastasis. And it is prevalent in a subtype of breast cancer, which is very aggressive. Therefore, PTP1B inhibition can produce tumor inhibition, and Dr. Tonks' lab at CSHL has been focusing on research on this application. This shows some of the data coming out of the CSHL labs on the trodostomine compound. These are xenografts of BT474 human breast cancer cells in mice. And as you can see, the control cells, control animals, when they are given these xenografts, produce substantially large tumors developing from the xenografts. The MSI-treated compound rats in the middle show the initial tumor implant, and the right-hand side photograph shows the tumor size after the treatment duration. The graph at the bottom shows the control cells, the control rats, showing a considerable large tumor volume, as is evidenced by the size of the tumors here, whereas the trodostomine-treated mice at the end of the trial had still small tumors, pretty much comparable to the initial xenograft. So the compound seems to have good tumor regression capabilities. More interesting, perhaps, is the effect on the lung metastases that are induced in HER2-positive mammary cancers. On the left, you can see a lung from a control animal with very substantial lung metastases being developed, whereas upon treatment with either a 5 milligram per kilo or a 10 milligram per kilo dose of trodostomine, the tumor volume in the treated animals is very, very low. So the metastases induced by the HER2 activation in these tumors can be essentially abrogated by MSI 1436 with the trodostomine. In obesity models in mice, again, the compound showed for weight loss in mice at doses ranging from 2 milligrams up to 5 milligrams, it produced a good dose-response effect. That shows that there's probably a dose response commensurate with the PTP1B inhibition producing the weight loss in these animals. Where we are currently with trodostomine, the parent compound, is we have already conducted a pre-IND meeting with the FDA. The IND for the phase 1 trial in breast cancer is to be submitted shortly. The phase 1 trial is designed as a single-agent, open-label dose escalation study to find potential effective doses. It is going to be a 20-patient trial with 20 patients with metastatic breast cancer. The clinical trial, obviously, is going to be at another well-known Long Island institution that you all know and love very much, and the initiation of the trial is expected later this year. The breast cancer market obviously is huge, with 60 to 75,000 patients having HER2 positive disease, and HER2 positive breast cancer is particularly aggressive and has a poor prognosis. The current market leaders in terms of drugs to address HER2 positive breast cancers are Herceptin, which is the antibody drug from Genentech, a large molecule antibody, expensive to produce relative to a small molecule, obviously. And Herceptin did $6.5 billion in annual sales internationally, and Tycub is the product from GlaxoSmithKline, which also has a substantial market. Our corporate strategy, therefore, is to conduct a phase 1 trial with trodostomine in breast cancer, and therefore validate PTP1B as a validated target for this indication. And concurrently, we are working on the next generation of trodostomine analogs, which have higher potency and potential oral bioavailability. Oral bioavailability would particularly be useful in an obesity or diabetes application of such PTP1B inhibitors. The current research is being conducted at CSHL by Dr. Thomas's laboratory. The long-term strategy is to license and monetize the portfolio after the clinical proof of concept is established in the clinical trial. The capital requirements, we are currently seeking in $1 to $2 million in seed equity financing. The use of funds is enumerated here. The trial is going to cost us in the neighborhood of $400K, and we are going to build out the research infrastructure as well. The current ownership is 50% CSHL, 50% all pharmaceutical. The value proposition is we're going to be in the clinic by the year end of this year. It is a novel approach targeting PTP1B. We are developing a library of more potent and or orally available analogs, and continuing the collaboration with Cold Spring Harbor in this partnership. Thank you very much.
Questions?
Yes.
Given what I perceive to be the obvious closeness, given your email and some of your comments about we at Or Pharmaceutical, why seek outside funding? Why not just make this a research project, internal research and development at Or?
Well, the Cold Spring Harbor has been a very effective partner, and Or, the parent company, has stated that it's going to develop a core ophthalmology focus. So we're in the process of partnering out the other programs that are not ophthalmology, and this fits in very well with that.
Thank you.
Any other questions? Okay, thank you very much.
Thank you.